Hi there, my name is Giselle Mendez and I will be starting my simulation lab. This is for the course Biochemistry and Molecular Biology. So this is my attempt. Start simulation. Um, this is the context. I have already have read all of this context, so I know what I will be doing for this lab, and I also have went over all the equipment. On the prediction, is a selected tube that you think best represents a successful transformation. So I think it's tube 4 since I can see the plasmids already inside. And then select the tube that you think represents negative control. The negative control states that your control group will contain a component equal as cells, but no recombinant plasmid. So the only one that does not have recombinant plasmid inside is this one. Now we start the simulation. Mm, First step, set the water bath to 42 degrees Celsius. Next step is pick up the 200 micropipette, set it. Use the volume setting to change to 50 UL. Save volume. Open the P200, P200, close the P200, open the CC tube, move over the 200 microp to the CC tube, drop 50 ULCC solution pushing out under first stop, open the P minus tube, dispense the CC to the P by holding down the plunger until it reaches the second stop. Now I have to close it and I just open the biohazard. It says to pick it up and put it back in the ice spot, and then you move open to the biohazard waste container. I already done that. Move the pipette over the biohazard and this eject. Now you repeat the same process again for the P plus tube. Open this. Close it. Open the CC. First stop. Open the T plus. Dispense to the second stop. Close the CC tube. Close the P plus tube. Open the biohazard waste. And eject tip. Put it back. Close. Now go to the next step, which is pick up the P20 micropipette. Use the volume setting to change to 10 UL. It's a 2, so we change it to 10. Oops. Now it says move to the P20. Come and touch the tip. Para tube. I have to close this and open the para tube over the para tube. Draw up. I need to open it first. So I draw up 10 UL by first stop think I already did it, right? Because it went. Now I opened the P plus tube and I put it in the P plus tube. Now mix the button, and that was where the problem started. Hey, let me close these. Doesn't give you the option to mix, so let's pretend like we mixed it. So we mixed it. Okay, so replace the P plus tube back into the ice bucket immediately. Put it in the ice bucket. Replace the P. Open the biohazard waste container, and then you eject the tip. And then back, put it back. Now it says set the timer for 15 minutes while the P plus and P are on ice. The timer for 15 minutes. Okay. I paused it. Man. Okay. Start. Close. Next, it says open the water bath lid. Opening it. Pick up the P minus and P plus tubes and place them into the floating tube rock. So you put the P minus and the P plus and the tube rock. Set the water timer for 45 seconds. Water bath timer for 45 seconds. For 45 seconds. Yeah, that's 45 minutes. 45 seconds, okay? And then it says pick up the floating tube rock and place it into the water bath. There. Close the water bath lid. Start the water bath timer. Oh shoot. I was supposed to. Okay, now you put the 45 and then you start it. And then it says open the water bath lid. Then you open it, move the P plus tubes back to the ice bucket. Okay. Okay. Now set the timer for 1 minute. 1 minute. 1 minute. And then just to set the timer because didn't say start it. So I'm just gonna set it. Next, it says pick up the P200 micropipette from the micropipette rock. Again, use the ones and change it volume to 150. Okay, 150. $150 UL. Open the P200 pipette and pick up the micropipette. So pick up the micropipette, place the micropipette so the tip is in the LV solution. Okay. In the LV solution. Draw up the LV by pressing the plunger down until it reaches the first stop and then slowly releasing it. One slow. Oh, I have to open the solution. I forgot that it needs to be opened. Okay. Now, yes, we can release we can suck up the liquid. Um, Open the P negative tube. Okay. Open. Dispense the LB into the P minus tube, holding down the plunger until it reaches the second stop. So now we're going to be releasing the right. Mix this to solution tubes using the pipette. Well, I tried to mix it the last time and it didn't work, but let's pretend it already mixed. Mix the solution tube using the pipette. Open the biohazard waste container and eject the tube, right? Move it back away and then use it. Yep. And now it says repeat the process to add 150 ml UL of LB to the P plus tube. So now, I should have closed this. It's contamination. But since it's just a simulation, it didn't get contaminated. But it should have been closed. Now I'm gonna open it again. It's supposed to be sterile. And it's already at 150 UL. So close it. And then in my LB, that should have been closed too. Everything should be closed right after using it. Soak up the liquid. Let's close it now. Close up the LB. Open the P plus, since we're doing it for the P plus now. And we release slowly. And voila. We close it because you have to close it, and we eject the tip and we put back our micropipette because they're very expensive. Set the timer for 15 minutes and incubate the cells at room temperature. Okay, set the timer for 15 minutes and incubate the cells at room temperature. So if it says incubate the cells at room temperature, that means we'll take it off the water bath because it's in an ice bucket and it's not room temperature. So I'll put it in the rock. My P minus and P plus. It says incubate the cells at room temperature. So then let's set the timer first. Well, let's set the timer at 15 minutes. 15 minutes. Okay. We start it and that's it. Then let's see what happens. Next, results. After you have performed your transformation experiment, you will not be able to confirm your results simply by looking at your tubes. The next experiment step is to place your bacteria on solid medium so that you can observe their growth. For the simulated environment, we have provided a hypothetical view of your results inside the tubes. So predicted results, actual results. P plus, P minus. Yeah, it says it's correct. Transformation was successful because some of the bacteria in the experimental group contains plasmids. The bacteria in control group should have no plasmids, and none were present in the tube. Below is a representation of the ideal results for a transformation experiment. Ideal results. For a successful transformation, the circular plasmid DNA needs to be taken up into the bacterial cells. The negative control contains components of E. coli cells, but no plasmid, so the bacterial cells cannot be transformed. Okay, yes, I get that. It's correct. Reflection. Well, let's look at the learned simulation. Please click on your right answer to each of the following questions to save them in your notebook. With what is a bacterial transformation? Well, a process through which the genetic makeup of a cell is altered by introducing foreign DNA into a bacterial cell. Okay, a process in which the genetic makeup of a cell is mutated by introducing new enzymes. Not mutated. A process in which dead bacterial cells take up DNA. I think it's A. Yeah. How does the P negative tube differ from the P plus tube? Well, has a control plasmid added to it that does not contain the gene of the ampicillin resistance of the. Well, P plus contains a plasmid with the gene ampicillin resistance. P minus contains the recombinant plasmid but no competent cells. So P negative contains the component cells but no recombinant plasmid, while P plus. I think it's C. I'm not sure. Oh, yes, it's C. Select whether the following statement is true or false. Lurea broth is primarily used to culture for bacteria. True is it is a good food source for the bacteria. False, it kills most bacteria. I think it's true because I mean it's a broth. I've never heard of Lurea, but it's broth. I know broth is used for bacteria. Use aseptic techniques during this lab. Why is it important to work in a sterile manner when working with the bacteria in the lab? Well, to prevent DNA contamination from the environment that may also then be transformed into the bacterial cells, to prevent the transfer of bacteria from the experiment to the environment and vice versa. Yes, that's true. You need to take care of the bacteria, or if it's a dangerous bacteria coming into the environment. Sterility is not important in this lab and type of just nonsense. Sterility is always important. So I think it's B. And 5, let's see. Why are the cells incubate at 42 degrees Celsius? Bacterial cells begin releasing DNA for other cells to take up. This is a good recovery temperature after heat shock. At 42 degrees, the members of the bacterial cells begin to degrade, creating pores through which DNA can enter the cell. Um, I really don't know the answer to this one, but let me see. At 42 degrees. I'm not sure. I think it's C. Makes more sense, but I'm not sure. Oh, it's correct. Weird. How confident do you feel about using the technique you learned? I am the using the technique you learned, like all of the. I am confident, somewhat confident, slightly confident, not confident. I am very confident. I am fairly confident because I know that I should have closed the micropipette. Sometimes I forgot to close it or close the tube that might have caused contamination. But I think I'm very content because I did follow the steps and it's just that. So I'm gonna say fairly content. Submit answer. Oh. Next section. Summary. In this simulation, you learn how to transfer bacteria using the heat shock. The simulation explores how to recap. Prepare bacterial cells and plasmid DNA mixtures for transformation. Set up the relevant controls for the transformation process and analysis. Perform heat shock, which temporarily causes pores in bacterial cells members to form, facilitating the uptake of external DNA, which is true. That's why the 42 degrees Celsius. Now I get it. Take away messages. When working in the bacteria, you need to be sterile. Blah blah blah. Bacteria, very very important. Bleach and alcohol are the the best to use for sterility. Transformation E. coli needs to be pre-treated with calcium and heat for more component take DNA more efficiently. Always be careful to mix component cells gently by pumping with the micropipette next. Never vortex competent cells. Ensure competent cells are kept on ice to create a temperature gradient that will increase the efficiency of transformation. Be conscious of heat transfer and ensure all tubes are handled at the rim when pipetting. Oh, see, we couldn't do that because it's a simulation, but it's good to know. Cells need time to recover after the heat shock so they can repair the holes in the cell membranes. Makes sense, right? So here we go, the end of my simulation. Thank you for watching. I hope it was entertaining.